Hello everyone. Thank you a lot for all the supports and valuable suggestions. Now today we are presenting a brief discussion on eukaryotic cell structures. As we discussed in our previous presentations, cell is the fundamental unit of life, and there are two types of organisms based on their cell structure. They are prokaryotes and eukaryotes. Today our main topic is eukaryotic cell and its components. Now what is eukaryotes? Eukaryotes are organisms whose cells have a nucleus within a nuclear envelope in which the well-defined chromosomes are located. Eukaryotes are containing organized organelles, among which some are enveloped by membrane and some are not, such as mitochondria, Golgi apparatus, endoplasmic reticulum, lysosomes, ribosomes, etc. etc. Eukaryotic cells are exclusively found in plants, animals, fungi, protozoa, and other complex organisms. This is a well-defined diagram of animal cell, and this is a diagram of plant cell, which is the typical examples of eukaryotic cells. Here we can observe a nucleus in both cells, which is well organized by nuclear envelope, and well-defined nucleus also observed. There are so many common organelles in animal cells and plant cells, as well as there are many differences also. The cytoplasm of plant cells surrounds by cell wall, which is absent in animal cell structure. In other hand, you can see animal cell contains centriole and plant cell don't. Plant cell also contain another double membrane bound organelle, which is called plastids. You can see here, this is called plastid, which is absent in animal cell. Now, as we said in our previous presentation, the cytoplasm of cell surrounds by different structures. Which are they? They are cell wall and cell membrane. As we know, cell wall is a non-living, rigid structure outside the cell membrane. It is found in plant cells and fungi. Basically, cell wall provides shape to the cell. The basic component of cell wall is cellulose, but there are so many components present in cell wall other than cellulose, such as hemicellulose, proteins, and pectin are present in plant cell wall, and in other hand, fungi cell wall has galactans, mannese, calcium carbonate, etc. etc. As we discussed before, the structure of cell wall has three layers: middle lamella, primary wall, secondary wall. The outermost layer, which adjoining two cells together, are called middle lamella. Here, this is called middle lamella. This layer after the middle lamella is called primary wall, which is made up of cellulose and hemicellulose. This layer is capable of growth, and this innermost layer is called the secondary wall. And this narrow thread-like structure of cytoplasm that passes through the cell walls of adjacent plant cells and allows communication between them is called plasmodesmata. This is the structure of cellulose. Cellulose is composed of a long chain of at least 500 glucose molecules. Cellulose is a polysaccharide. Polysaccharide is a Latin word which stands for many sugars. In other hand, fungal cell walls are rigid and contain complex polysaccharide called chitin. Chitin adds structural strength to its cell wall. Other major constituent of fungal cell wall is glucan. Here, chitin is a structurally important component of fungal cell wall, which located closest to the plasma membrane. Here you can see, this is the cell membrane of a fungal cell, and the uppermost layer is made up with chitin. As we know, the main function of cell wall is providing shape to the cell. Besides that, cell wall protects cell from injury, pathogens, and osmotic shock also. It also regulates the transportation of substance into and out of cells. After cell wall, we discussed about another structure which found in all cells basically, that separates the interior of the cell from the outside environment is called cytoplasmic membrane, also called the cell membrane or plasma membrane. In plant cells, plasma membrane is attached after cell wall on its inner surface. As we discussed in our previous presentation, phospholipids make up the basic structure of a cell membrane. A single phospholipid molecule has two different ends, a head and a tail. The tail is basically made up with polar and hydrophilic components. Hydrophilic means water loving. For that reason, heads face the outside and inside of the cell. These heads interact with the aqueous environment outside and within a cell, because it's made up with hydrophilic components. And the tail basically made up by non-polar hydrophobic components. Hydrophobic means water repelling. The tails are sandwiched between the heads like this, and they are protected from the aqueous environment. You can see here the structure. Those are the hydrophobic tails which are sandwiched like this. This arrangement of phospholipid molecules makes up the lipid bilayer structure. The bilayer structure looks like a mosaic and has a semi-fluid nature. The lipid bilayer gives the fluidity and elasticity to the membrane, in which the integral proteins are embedded and looks like the proteins are floating in lipid bilayer. Here those are the peripheral proteins are associated loosely with the inner membrane surface. The small spheres represent the hydrophilic ends, and the wiggy tails represent the hydrophobic fatty acid chains. Singer and Nicholson named this structure fluid mosaic model, which is the most widely accepted model of cell membrane. Now, what is the function of cell membrane? As the plasma membrane is selectively permeable, we know it. In our previous presentation, we discussed about it. It allows only selected substances to pass through. So the basic job of cell membrane is allowing transport of molecules across the membrane. In our last presentation, we discussed the transport system by cell membrane in detail. If you want to know, you can follow our last presentation. There are two types of transport system. One is active transport, which occurs against concentration gradient, and one is passive transport, which occurs along with concentration gradient. Now, here you can see sodium-potassium pump is a simple example of active transport. The sodium-potassium pump moves sodium ions out of the cell and potassium ions into the cell. It moves two potassium ions into the cell where potassium levels are high. That means it transports in the high concentration from the low concentration, and pumps 3 sodium ions out of cell and into the extracellular fluid. Now 3 sodium ions bind with this carrier protein. This is the carrier protein which pumped inside the cell. The carrier proteins gets energy from ATP. It pumps the 3 sodium ions out of the cell. At that point, 2 potassium ions from outside the cell bind to the protein pump. Then the potassium ions are transported into the cell and the process repeats. Now, passive transport does not require any energy input. As we know, this movement of molecules from an area of high concentration to an area of low concentration is called diffusion. Here you can see this is the low concentration area, this is the high concentration area, and the molecules move towards the high to low. This is example of passive transport. This is a simple diffusion. This is called simple diffusion. If this simple process carried out by a carrier protein or channel proteins, this is called facilitated diffusion. Now, what is after the cell membrane? Inside the cell membrane, a clear, thick liquid that fills the cell is called the cytoplasm. In eukaryotic cells, the cytoplasm includes all of the material inside the cell and outside of the nucleus. The cytoplasm is about 80% of water and as usually colorless. The main component of cytoplasm are cytosol, the organelles, and the cytoplasmic inclusions. Most cellular activities take place within the cytoplasm, such as metabolic pathways, including glycolysis, and many processes such as cell division. Inside the cell, the concentrated inner area of cytoplasm is called the endoplasm, and the outer layer near the cell membrane is called the ectoplasm. Now, talk about the constituents of cytoplasm. At first, we discuss about cytosol. Cytosol is a gel-like substance. The cytosol is the part of the cytoplasm that is not occupied by any organelle. Cytosol makes up about 70% of the cell volume and is a complex mixture of cytoskeleton filaments, dissolved molecules, and water. Now, what is cytoskeleton filaments? The cytoskeleton is the network of fibers forming the eukaryotic cells. It mainly provides the shape and support to the cell. In eukaryotes, it is composed of three main components, which are microfilaments, microtubules, and intermediate filaments. Now you can see in this diagram, this is microfilament. This is also known as actin filaments, are composed of linear polymers of G-actin proteins, and this is responsible for muscle contraction. These filaments are intermediate filaments, averaging 10 nanometer in diameter. They are more stable than microfilaments. Basically, intermediate filaments organize the tridimensional structure of the cell, the 3D structure of the cell. And these are the microtubules. Microtubules are hollow cylinders about 23 nanometer in diameter and most commonly comprising 13 protofilaments that in turn are polymers of alpha and beta tubulin. Microtubules are very dynamic behavior. They keep growing and shrinking steadily. These help in transporting cellular materials and dividing chromosomes during cell division. Now the other constituent of cytoplasm is the organelles. Those are the main components, I think, of the cytoplasm. According to the presence of membrane, organelles are three types, such as non-membrane organelles, single membrane bound organelles, and double membrane bound organelles. Now, non-membrane such as ribosome, single membrane such as lysosome, Golgi apparatus, endoplasmic reticulum, double membrane such as nucleus, plastid, mitochondria. Now, at first we are going through the double membrane bound organelles. As we said before, double membrane organelles are nucleus, mitochondria, plastid. Now, nucleus is the main organelle of a cell. It is called the brain of a cell. The nucleus is a membrane-bound organelle that contains genetic material of eukaryotic organisms, which is DNA, deoxyribonucleic acid. Nucleus serves to maintain the integrity of the cell by facilitating transcription and replication processes. Nucleus is the largest organelle inside the cell, taking up about a tenth of the entire cell volume. The main structures making up the nucleus are the nuclear envelope, a double membrane that encloses the entire organelle and isolates its contents from the cellular cytoplasm. A network within the nucleus that adds mechanical support, much like the cytoskeleton. As the nuclear envelope is impermeable to large molecules, nuclear pores are required to regulate nuclear transport of molecules across the envelope. The interior of the nucleus does not contain any membrane-bound subcompartments, such as nucleolus, which is mainly involved in the assembly of ribosomes. After being produced in the nucleolus, ribosomes are exported to the cytoplasm, where they translate mRNA. The contents of the nucleus are held in the nucleoplasm, similar to the cytoplasm in the rest of the cell. The fluid component of this is termed the nucleosome. The cell nucleus contains the majority of the cell's genetic material in the form of multiple linear DNA molecules organized into structures called chromosomes. Each human cell contains roughly 2 meters of DNA. During most of the cell cycle, these are organized in a DNA-protein complex known as chromatin. And during cell division, the chromatin can be seen to form the well-defined chromosome. There are two types of chromatin. Euchromatin, which is less compact DNA form and contains genes that are frequently expressed by the cell. The other type is heterochromatin, which is the more compact form and contains DNA that is infrequently transcribed. Chromosomes have certain constriction sites called centromeres. Based on the position of the centromere, chromosome can be divided in 4 types: metacentric, submetacentric, acrocentric, and telocentric. What is metacentric? Metacentric is with centromere in the center and having equal chromosome arm. And in submetacentric, centromere is slightly off-center, creating one short and one long arm. And in acrocentric, centromere is extremely off-center with one very long and one very short chromosome arm. And for telocentric, centromere is placed at one end of the chromosome. Humans do not possess telocentric chromosomes. Now, what is the function of nucleus? The main function of the cell nucleus is to control gene expression and mediate the replication of DNA during the cell cycle. It has been found that replication happens in a localized way in the cell nucleus. Gene expression first involves transcription, in which DNA is used as a template to produce RNA. In the case of genes encoding proteins, that RNA produced from this process is messenger RNA, which then needs to be translated by ribosomes to form a protein. As ribosomes are located outside the nucleus, mRNA produced needs to be exported. Since the nucleus is the site of transcription, it also contains a very variety of proteins that either directly mediate transcription or are involved in regulating process. And the other functions of nucleus is transporting various molecules in and out of the nucleus through the nuclear pores. And during cell division, the nucleus is integrated and disintegrated to divide the contents and the genetic material. The cell nucleus is also associated with programmed cell death or apoptosis. And in nucleus, newly synthesized mRNA molecules are processed. The newly synthesized mRNA are known as primary transcript or pre-mRNA. They must undergo post-transcriptional modification in the nucleus before being exported to the cytoplasm. mRNA that appears in the cytoplasm without these modifications is degraded rather than used for protein translation. So, the processing of pre-mRNA is a very important function of nucleus. Now we will discuss about another double membrane bound organelle which is found in all of eukaryotic organisms called mitochondria. Mitochondria is a plural term. The singular term is mitochondrion. Now mitochondria is also known as powerhouse of the cell. Some cells may lack mitochondria or mitochondria. We can use mitochondria. Some cells may lack mitochondria, such as mature mammalian red blood cells. The number of mitochondria in a cell vary widely by organism, tissue, and cell type. For instance, red blood cells have no mitochondria, whereas liver cells have more than 2000 of mitochondria. The organelle is composed of compartments that carry out specialized functions. These compartments or regions include the outer membrane, the intermembrane space, the inner membrane, and the cristae and matrix. Now, here we can see a diagram of mitochondrion. A mitochondrion contains outer and inner membranes composed of phospholipid bilayers and proteins. The two membranes have different properties. Because of this double membrane organization, there are 5 distinct parts of mitochondria. They are the outer mitochondrial membrane. This is the outer mitochondrial membrane. The intermembrane space. The intermembrane space is this space. The space between the outer membrane and the inner membrane is called the intermembrane space. And this is the intermitochondrial membrane. This is the intermitochondrial membrane, and those are called the cristae. Cristae is formed by infoldings of the inner membrane. And this is the matrix. Matrix is the space within the inner membrane. The inner mitochondrial membrane is compartmentalized into numerous cristae, which expand the surface area of the inner mitochondrial membrane and enhancing its ability to produce ATP. For typical liver mitochondria, the area of the inner membrane is about 5 times as large as the outer membrane. This is just because the enhance the ability to produce the ATP. As we know, most of a cell's DNA is contained in the cell nucleus. The mitochondrion has its own independent genome. Here you can see, which is known as mitochondrial DNA. The mitochondrial proteins also transcribed from this mitochondrial DNA. Those are called, the genome of mitochondria is called mitogenome. Now, what is the function of mitochondria? The most prominent roles of mitochondria are to produce the energy currency of cell, which is ATP, through respiration and to regulate cellular metabolism. The central set of reactions involved in ATP production are collectively known as the citric acid cycle or the Krebs cycle. A dominant role for the mitochondria is the production of ATP as reflected by the large number of proteins in the inner membrane for this task. This is done by oxidizing the major products of glucose, pyruvate and NADH, which are produced in the cytosol. This type of cellular respiration, known as aerobic respiration, is dependent on the presence of oxygen, which provides most of the energy released. The production of ATP from glucose. The mitochondria has many other functions in addition to the production of ATP, such as signaling to mitochondrial reactive oxygen species, regulation of membrane potential, apoptosis. Apoptosis is programmed cell death, calcium signaling, regulation of cellular metabolism, steroid synthesis, immune signaling, etc., etc. Now we will talk about plastid, which is a double membrane organelle found in the cells of plants, algae, and some other eukaryotic organisms. Plastids are the site of manufacture and storage of important chemical compounds which used by the cells of autotrophic eukaryotes. They often contain pigments used in photosynthesis, and the types of pigments in a plastid determine the cell's color. In plants, plastids may differentiate into several forms such as chloroplasts, chromoplasts, and leucoplasts. Chloroplasts are green plastids for photosynthesis. These plastids contain chlorophyll. Chromoplasts are colored plastids for pigment synthesis and storage, and leucoplasts are colorless plastids. Now, this is the structure of chloroplast. All chloroplasts have at least 3 membrane systems: the outer chloroplast membrane, the inner chloroplast membrane, and the thylakoid membrane. The outside of the thylakoid space is called the stroma. This is the plastoglobule, which consists of lipids and proteins. These are the thylakoid system and the collection of membranous sacs called the thylakoids, where chlorophyll is found, and the light reaction of photosynthesis happens here. And the thylakoids are arranged in stacks are called grana. Now, what is the function of chloroplast? Chloroplasts trap the solar energy and manufacture food through photosynthesis, and chromoplasts provide color to various flowering parts. Because chloroplasts contain so many pigments such as yellow pigment, red or orange, etc. etc. Keratin is an orange pigment, xanthophyll is a yellow pigment. Those are the examples of chromoplasts. Now, chloroplasts are colorless pigments, but chloroplasts can store many molecules such as carbohydrates, oil, fats and proteins. Leucoplasts are colorless, but they can store carbohydrates, oils and proteins. Amyloplasts store carbohydrates, elaioplasts store oil and fats, and aleuroplasts store proteins. Now we will going through the single membrane bound organelles. Single membrane bound organelles are Golgi apparatus, endoplasmic reticulum and lysosomes. The Golgi apparatus, also known as the Golgi complex, Golgi body or simple the Golgi. These organelles found in most eukaryotic cells. This organelle was named after its discoverer. In 1897, Italian scientist Camillo Golgi identified this organelle. In most eukaryotes, the Golgi apparatus is made up of a series of compartments and is a collection of fused, flattened membrane enclosed discs known as cisternae, which are originating from vesicular clusters that bud off the endoplasmic reticulum. It is present in all eukaryotic cells except human red blood cells and sheep cells of plants. Golgi apparatus has two phases, cis entry phase and trans-exit phase. Cis phase receives vesicles from endoplasmic reticulum. These vesicles fuse with Golgi apparatus and form new cisternae. Vesicles filled with processed proteins and lipids and then exist from the trans phase of the Golgi apparatus. Now, what are the functions of Golgi apparatus? Golgi apparatus is a major collection and dispatch station of protein products received from the endoplasmic reticulum. Proteins synthesized in endoplasmic reticulum and packaged into vesicles, which then fuse with the Golgi apparatus. These carboproteins are modified and destined for secretion via exocytosis or for use in the cell. In this respect, the Golgi can be thought of as similar to a post office. It packages and labels items which it then sends to different parts of the cell or to the extracellular space. The Golgi apparatus is also involved in lipid transport and lysosome formation. Our next organelle is endoplasmic reticulum. The endoplasmic reticulum is found in most eukaryotic cells and forms an interconnected network of flattened membrane enclosed sacs and tubular structures. The membranes of endoplasmic reticulum are continuous with the nuclear membrane. The endoplasmic reticulum is made up of two subunits. One is rough endoplasmic reticulum and one is smooth endoplasmic reticulum or ACR. The endoplasmic reticulum is not found in red blood cells. The outer face of the rough endoplasmic reticulum is studded with ribosomes that are the sites of protein synthesis, and the smooth endoplasmic reticulum lacks ribosomes and functions in lipid synthesis, the production of steroid hormones and detoxification. The smooth endoplasmic reticulum is especially abundant in mammalian liver and gonad cell. Now, this is the diagram of a endoplasmic reticulum. The surface of rough endoplasmic reticulum is studded with protein manufacturing ribosomes, giving it a rough appearance. You can see the presence of ribosome. Those are the ribosomes, and these are called smooth endoplasmic reticulum because there is no ribosome. The membrane of the rough endoplasmic reticulum located near and continuous with the outer layer of nuclear envelope. Now, what are the functions of endoplasmic reticulum? The endoplasmic reticulum serves many general functions, including the folding of protein molecules in sacs called cisternae and the transport of synthesized proteins in vesicles to the Golgi apparatus. The folding of newly made proteins is made possible by several endoplasmic reticulum chaperones proteins. Because only properly folded proteins are transported from the rough endoplasmic reticulum to the Golgi apparatus. Unfolded proteins causes an unfolded protein response as a stress response in the endoplasmic reticulum. And the functions of smooth endoplasmic reticulum, such as smooth endoplasmic reticulum is responsible for the synthesis of essential lipids such as phospholipids, cholesterol. Smooth endoplasmic reticulum is also responsible for the production of steroid hormones. And smooth endoplasmic reticulum store and release calcium ions, which are quite important for the nervous system and the muscular system. Now we will discuss about lysosome. Lysosome is a membrane-bound organelle found in many animal cells. They are a spherical vesicle that contain hydrolytic enzymes that can break down many kinds of biomolecules. A lysosome has a specific composition of both its membrane proteins and its luminal proteins. The optimal pH of lysosome is less than 7. Besides degradation of polymers, the lysosome is involved in various cell processes, including secretion, plasma membrane repair, apoptosis, cell signaling, energy metabolism, etc., etc. Lysosomes act as the waste disposal system of the cell by digesting unused materials in the cytoplasm from both inside and outside the cell. Materials from outside the cell taken up through endocytosis, and the material from the inside of the cell is digested through autophagy. Enzymes of the lysosomes are synthesized in the rough endoplasmic reticulum and exported to the Golgi apparatus. The enzymes are trafficked from the Golgi apparatus to lysosomes in small vesicles which fuse with larger acidic vesicles. Now, what is the functions of lysosomes? The great function of lysosomes is to degrade or break down macromolecules. This includes processing old and worn-out cell parts to recycle their components and making harmful toxins or bacteria safe by degrading them. They process many of the vacuoles which move either in or out of the cell, ensuring things run smoothly. Lysosomes are essentially the garbage disposal or recycling units of cell. In this sense, lysosomes have many different functions, including handling exocytosis, autophagy, heterophagy, and biosynthesis. Now we will discuss the non-membrane organelle, that means ribosome. Today we will discuss only about eukaryotic ribosome. Ribosomes are a large and complex molecular machine that catalyzes the synthesis of proteins referred to as translation. The ribosome selects amino-acylated transfer RNAs, which means tRNAs, based on the sequence of a protein encoding messenger RNA, that means mRNA, and covalently links the amino acids into a polypeptide chain. The ribosomes of eukaryotes are much larger than prokaryotic ribosomes and subject to more complex regulation and biogenesis pathways. Eukaryotic ribosomes are also known as ATS ribosomes, referring to their sedimentation coefficients in Weisberg unit. What is Vedvarg unit? The A stands for Vedvarg. And what is sedimentation coefficient? The sedimentation coefficient of a particle characterizes its sedimentation during centrifugation. It is defined as the ratio of a particle's sedimentation velocity to the applied acceleration causing the sedimentation. This is called the sedimentation coefficient. And the unit of sedimentation coefficient is Vedvarg. As the eukaryotic ribosome sediment faster than the prokaryote, for that reason they are 80th and prokaryotic ribosomes are 70th. Eukaryotic ribosomes have two unequal subunits designated as small subunit 40th and large subunit 60th according to their sedimentation coefficients. The small subunit monitors the complementarity between tRNA anticodon and mRNA, while the large subunit catalyzes peptide bond formation. Now, what is the function of ribosome? The function of a ribosome in any cell is to produce proteins. Proteins are used in almost all cellular functions. As catalysts, they speed the time of reactions. As fibers, they provide supports, and many protein functions in specific tasks. Recognizing the structure of the mRNA bound to a tRNA, the subunits of ribosome can combine to start synthesizing protein from the mRNA strand. The ribosome acts as a large catalyst, forming peptide bonds between amino acids. The used tRNA is released back into the cytosol so that it can bind to another amino acid. Eventually, the mRNA will present a codon to the ribosome, that means stop. And then the process will end it. Protein will detach the string of amino acids from the last tRNA and the protein will be released. At the last, we will discuss about a vacuole. A vacuole is a membrane-bound organelle which is present in all plant and fungal cells and in animal cells also. Vacuoles are essentially enclosed compartments which are filled with water-containing inorganic and organic molecules, including enzymes in solution. True, in certain cases, they may contain solids which have been engulfed. Vacuoles are formed by the fusion of multiple membrane vesicles and are effectively just larger forms of these. The organelle has no basic shape or size. Its structure varies according to the requirements of the cell. Here, this is a central vacuole of plant cell. The vacuole surrounded by a membrane called tonoplast. This area is called the tonoplast. This structure we can observe in plant cell. Now, what are the functions of vacuole? In general, vacuoles are isolating materials that might be harmful or a threat to the cell. It containing waste products, containing water in plant cells, maintaining the internal hydrostatic pressure or turgor in the cell, maintaining the acidic internal pH, exporting unwanted substance from the cells. Now that's it for today. In our next presentation, we will present the cell cycle and the cell division and the types of cell division. If you like our presentation, then please like and share our content and subscribe our channel. Thank you.